week of June 14th. And for that course, it would be 3 lectures per week with 1 to 2 office hours per week. So you guys can come in and ask any other questions related to the lecture or not, to aid you guys with the MCAT studying. And we aim to, cover most of the high yield MCAT content that you will be expected to know, at least for the exam. And I also forgot to mention, Happy Mother's Day, everyone. So if you haven't already, please, greet your mothers. And I'm going to try to keep this lesson relatively short this week, compared to the last few weeks, I guess. So our learning objectives this week are to review the major organelles and their functions in a eukaryotic cell, also to understand the endosymbiotic theory, and to review the structure of the cellular membrane, and following that, to understand the major distinctions between a prokaryotic and eukaryotic cell. And if you guys have a biology background, this may be quite a review for you guys. So yeah. I just wanted to start off first with, the basics of what is an organelle. And so, as seen in this bullet point, an organelle is a small membrane-bound structure within a cell that carries out specific cellular functions. Now, this membrane is usually made out of phospholipids. And the way I like to think of an organelle, I guess, within a cell, they are different compartments within the overall cell. So you could compare that even to like an amusement park, right? You have this whole park, and for each of the different rides, there's like a different section where specifically this thing is going on for each of those rides. So the different areas. And the major animal cell organelles that we will be going over today are going to be the nucleus, the mitochondria, the rough ER, the smooth ER, the Golgi Apparatus, lysosomes, and peroxisomes. I realize that I made a bit of a typo here because the ribosomes we will be going over in relation to the rough endoplasmic reticulum, but please remember that the ribosomes themselves are not considered another organelle. And so because they're not bound around by a membrane. And as you can see in the lower right-hand corner, there is also a diagram just giving an overview of all of the different organelles in relation to each other within an animal cell. And so the first organelle that I wanted to go over was the nucleus. And the nucleus, as many of you may know, is very important because that's where DNA is stored. It's stored and protected in the cell's nucleus. And within the nucleus, there is also the nucleolus that is most responsible for producing ribosomes. The unique aspect of the nucleolus also is that it does not have a membrane surrounding it. So that is also another structure that is not considered an organelle because of the fact that it doesn't have that membrane surrounding it. Specifically for the nucleus, though, that is surrounded by a nuclear envelope. And as you can see in the diagram to the right, the nuclear envelope is made up of an inner membrane and an outer membrane. You can see in the, I would say, blue-green color lines right outside where I'm pointing to, if you guys can see the pointer. And with it, like embedded in the nuclear envelope are nuclear pores that allow for the entry and exit of different molecules. Now, usually in processes like making mature mRNA, in the nucleus, you would have the DNA being replicated, and there would be transcription occurring to make the premature mRNA. And it gets processed, And after it gets processed and becomes the mature mRNA, that's how it leaves the nucleus via these nuclear pores. And usually the regulation for what enters and what exits the nucleus is based on a lot of cellular signaling. So that's a little bit more complex. There are different types of signaling molecules that we would most likely be going over in future lectures, especially the more high yield ones. And going on from the nucleus, as you can see in the left hand side, the picture here. So from the nucleus, it goes kind of directly to the endoplasmic reticulum. And so the endoplasmic reticulum is a very folded structure, but it consists of two parts. So the rough endoplasmic reticulum and the smooth endoplasmic reticulum. The rough endoplasmic reticulum is identifiable because it has these ribosomes being attached, as you can see on this left-hand side. And under a microscope, this is how scientists distinguish between the two, is because they saw this different rough-looking aspect of the RER. And the rough endoplasmic reticulum is most important because it's the site of protein synthesis. So after you get the mature mRNA, that's when it is further processed and made into that translated protein that gets partially modified while in the rough endoplasmic reticulum, and made into the 3D structure, like tertiary structure, based on the amino acid sequence. And then there's also the other part of the endoplasmic reticulum, which is the smooth ER. And the main functions that I wanted to share with you today are listed here on the right-hand side. So the smooth ER is mainly responsible for synthesizing lipids. So you could think of steroid hormones as an example of fats. And it also metabolizes carbohydrates and also plays a role in the detoxification of drugs and toxins. I see here's a chat question. Oh, from Opal. So how does a protein know whether to go to the endoplasmic reticulum versus cytosol versus Golgi after processing? Are there signals that tells it where to go? Yes, Opal, there are different cellular signals that, directs that is attached to the protein to direct it on where to go. But usually, so the protein would be made in the rough endoplasmic reticulum, and I will actually go into, what usually leads it to go into the Golgi in the next slide. Sorry, so Nusha Kaldar, where is this located, as in the endoplasmic reticulum, or which thing? Sorry, if you could be more specific. Oh, is the endoplasmic reticulum, like where is the endoplasmic reticulum? If that's the question that you're asking, yes, the endoplasmic reticulum is in the cell. So let me just track back a little bit. Sorry. So here in the bottom right corner, you can see the endoplasmic reticulum is this like dark blue-green area here on the left and also on the right side. And so all of this is taking place within the cell. And I just started off with the nucleus to start from the center, and then we're going to be going outwards from there. So hopefully that helps. So yes, we just ended with talking about the endoplasmic reticulum. And from the rough endoplasmic reticulum, as you can see in this Golgi apparatus diagram, which I'm going into next. You know, just going back a little bit from before, we were from the nucleus, and then we went to the endoplasmic reticulum. And specifically for what goes into the Golgi apparatus, you want to focus on the rough endoplasmic reticulum for that, because what actually, so to address Opal's question, what actually causes the rough endoplasmic reticulum to go over to the Golgi apparatus is that usually there's a signal that brings it there, and in the Golgi apparatus, it specifically gets glycosylated. So when it gets glycosylated, it is specifically targeted for the Golgi apparatus. And so just to go into what the Golgi apparatus is, so it's primarily found near the endoplasmic reticulum because of this close relationship in how the signaling of the molecules go through. And it's a set of stacked sacs. But the main function really in the Golgi apparatus is to, get these proteins and determine where to send them. So they sort and send proteins to their proper destinations. Sorry, can you send this PDF so we can have it? Oh, yes, I will be sending the PowerPoints as Mark usually does after the session is done, and also the recording will be sent via the GroupMe or another Socratic Med outlet, which we will notify you guys of there. And so, yes, the Golgi apparatus also continues modifying proteins made from the rough endoplasmic reticulum. It can, add phosphate groups, sulfate groups, and, carbohydrates to modify it for the next step, which I will, oh, sorry, is there another? Oh, yeah, sorry. So the next step I wanted to go into actually here is a preview from the Golgi apparatus. Usually vesicles are formed, and they then go over to the lysosome. And so what the lysosome does, the main function of the lysosome, you can see this is from the rough ER to the Golgi, and then the lysosome on the left-hand diagram. The main function is that they digest various molecules with specific enzymes called acid hydrolases. And acid hydrolases, they are specifically found just within the lysosomes. And the reason is because actually the pH within the lysosomes is different from the entire cell. And so the pH within the lysosomes is a pH of 5 compared to the entire cell, which has a pH of 7.4. And this specific, more acidic environment actually allows these enzymes to work specifically only in the lysosomes. And what they do is, they help digest and break down substances, usually proteins, that end up becoming released into the cytoplasm. And they usually break these proteins down into the building blocks. So the basic parts of these more complex proteins, so that the cell can end up using, these building blocks to create new material or create, new proteins, and reuse and recycle these digested materials. And lysosomes perform both autophagy, which is defined as self-eating. And so what that means is that it breaks down organelles, or like this cell's contents or other cells' contents within the body, and breaks down those contents in the building blocks so it can be reused. And the second thing is crinophagy, which is when lysosomes break down excess contents that the cell creates sometimes, when, I guess you could relate it to, excess secreted hormones that the cell would produce, that would be broken down after the cell realizes that it doesn't quite need as much. And then those building blocks for that could be used for some other purpose within the cell. And I just wanted to also mention that the reason why I went through the endoplasmic reticulum, then the Golgi apparatus, and then the lysosomes is because usually the MCAT has I feel like usually they ask a question related to that pathway in specific. And so that's considered the secretory pathway, going from the endoplasmic reticulum to the Golgi and to the lysosomes. And those are considered also the endomembrane system, because it's related to, what goes from through these organelles and could lead to, where they go, whether it be just within the cell, or also, products that could go outside of the cell or embedded in the cell membrane. And the other organelle that I wanted to also talk about is the peroxisomes. And so their main function is to isolate hydrogen peroxide and use catalase specific enzyme to break down hydrogen peroxide into something less damaging to the cell. And so usually peroxisomes break down, hydrogen peroxide into water and oxygen, so that it helps protect the cell. But peroxisomes also, break down reactive oxygen species, which can come out from fatty oxidation processes that happen in the cell. And so this organelle is really there to protect the cell from any damaging remnants or anything that happens through other processes. Yep. And so I just wanted to start with the first question. I will give you about 2 to 3 minutes to read and answer. I believe, Tim, that we'll have a poll for this one.
Yeah, the poll's uploaded. So okay. Poll in progress.
Okay, thank you.
All right.
And does it seem like everyone filled out the poll so far?
Yeah, we have most people filling it out. So yeah, everyone has said choice D.
Everyone has said choice D. So
yeah, 100%.
That's great. Let me just Yes, so all of you guys are correct. The answer is lysosome. Yeah, because specifically in this question, right, we can see that the gangliosides are not properly digested and instead accumulate in the cells. And we know that lysosomes are responsible for digesting these. So great job, everyone. We have actually 2 more questions. So I'm going to give you again 2 to 3 minutes for this question as well. Okay, how is the poll so far?
Yeah, so a few less people answered this time around, but we have
Should I give a little more?
no, I mean I think 2 minutes is enough time. So yeah, 75% of people chose A, 25% chose C.
Okay. So I'm just gonna do this. So the majority of the students got it right. So it is the Golgi apparatus. I will explain just how come it is specifically the Golgi apparatus then for this question. So the question says inclusion cell disease is a congenital condition that comes from the lack of a single protein. On an intracellular level, proteins meant for the lysosome are sent to the extracellular space. And considering this phenotype, which organelle is most directly affected by the missing protein? And we see here in this particular question that in the second sentence, it mentions that proteins meant for the lysosome are sent to the extracellular space, just a different location from where it's meant for. And if we track back to think of what the Golgi apparatus's function is, it is to, receive these proteins and sort and direct them to where they should be going after after receiving it from the rough endoplasmic reticulum to another location, whether it be the lysosomes or outside. And so that's why the Golgi apparatus is the correct answer in here, because that means that there is a problem with the Golgi apparatus, which is why it's not going to the right location. Okay, so it seems like, okay, that makes sense. So we're gonna go into the next question then. and also another 2 to 3 minutes for this.
All right, it looks like most people voted. Yeah, so most votes, 60% came in for D, 20% came for A and B.
20% for A, 20% for B, and 60% for D.
Yeah.
Okay. Well, here I'm gonna reveal the answer. The majority of you guys are correct. I'm also gonna go in to explain why A and B, and also C are maybe not the right answer in this case, but so the question's asking the function of which cellular structure would be least affected by a malfunctioning Golgi apparatus. And the way I would explain, so as I was going through this presentation, just it was recognized that it was in like a stepwise manner, right? So from the endoplasmic reticulum to, well, from the nucleus to the endoplasmic reticulum to the Golgi apparatus, and then to the following areas where it could go, right? And so in this question, you know, because we're asking which one would be least, and I'm just going to stress least affected by a malfunctioning Golgi, would be that anything that happened before to get to the point of the Golgi would probably not really affect, would probably not really be affected by a problematic Golgi apparatus, right? Because usually when a Golgi apparatus is malfunctioning or a certain process within a series of events, usually the processes that happen before that particular malfunctioning event would be fine. Up until that point where there is this malfunctioning point, every step afterwards would likely be affected. And so for this, because the secretory vesicles come from the Golgi, and it could end up either in the plasma membrane or the lysosomes. And so that's why those A, B, and C are steps that happen after. And so they would be most affected as opposed to least affected in this question. Does that make sense to everyone? Okay. Yes, and if you guys have any questions also, yeah, feel free to send a message. Oh, sorry. You kind of didn't understand. Okay. I guess, could I ask which part seemed a little confusing? or why would the answer be endoplasmic reticulum? Because the endoplasmic, okay, yeah, that's okay. Because the endoplasmic reticulum is the step right before the Golgi apparatus. And so the question is asking which structure would be least affected by a malfunctioning Golgi apparatus. And so because the endoplasmic reticulum happens first before the Golgi apparatus, if the Golgi apparatus is malfunctioning, it's not really causing any problem backwards to the endoplasmic reticulum. Usually the way the process of the secretory pathway goes is from the nucleus to the endoplasmic reticulum and then to the Golgi, and everything after the Golgi would be either secretory vesicle, the lysosomes, or the plasma membrane. Does that help? Okay, great. It's good to hear. Okay, and I'm sorry. Let me just how wouldn't the plasma be affected? So it would be affected. The plasma membrane would be affected. For this particular question, it's asking which one would be least affected. So both all answer choices A, B, and C would be majorly affected by the Golgi apparatus. Sorry, does that answer the question, Kaylin? Or are you asking like how would the plasma membrane be affected? You meant what? Okay. So how would the plasma membrane be affected? So the function of the Golgi, right, is to sort the proteins that it receives. And so the different locations that the contents from the Golgi apparatus could go is it would go through a vesicle, and this vesicle can either go to another organelle, which is the lysosomes, or it can actually be targeted for secretion, in which case it could, not because it could be targeted for secretion, it could still go in the lysosomes, but for the, usually for the vesicles, they either go to the lysosomes or they go either on the plasma membrane itself, so they could be embedded as another protein, which I will go more into in the following slides as to the different proteins that are on the plasma membrane, or it can actually be secreted outside of the cell, past the plasma membrane to the extracellular environment. And so because the Golgi would be malfunctioning in this case, it could affect the process of where it's actually supposed to go. Does that help with the understanding? Yes. Okay, great. So now we will, if there are no other, oh wait, let me just double check. Okay, yeah, Mark was explaining the process of the chain of events from the ER Golgi and then to other molecules. So now we are going to move on to the next organelle, the mitochondria, also known as the powerhouse of the cell because it's the main producer of ATP. And ATP is produced via cellular respiration. It goes through this would probably be a topic that would be gone in more in depth in a biochemistry unit, just for like the processes of biochemistry of glycolysis, the citric acid cycle, and the electron transport chain. But each of these different processes happen also in different areas within the mitochondria in order to produce the ATP. But the main thing that I want you to focus on right now, at least for this lecture, is just mitochondria the main producer of ATP, energy for the cell. And the endosymbiotic theory is the one that is associated with the mitochondria. And so what this theory was saying is that mitochondria were once prokaryotic cells that were engulfed by larger cells in the past. And if you break down endosymbiotic, so endo means inner, within. And so when you think of these mitochondria being engulfed with another cell, the mitochondria are like within the other larger cell, which would be our normal eukaryotic cells in that case, because, you know, the organelle is like within the entire cell. And symbiotic, so having a symbiotic relationship where they both benefit each other. And so in that case, you know, mitochondria here are an organelle within the larger cell that's helping to create this energy so that the cell can use it. And so that's the endosymbiotic theory. And the other ways that scientists pointed to mitochondria perhaps potentially being prokaryotic in the past was because of the fact that they have their own genome. And so they have actually mitochondrial DNA as shown in the diagram to the right. I'm circling it. And this DNA in particular is actually like circular DNA. And so when you're thinking of prokaryotic cells, usually their DNA is in this circular form because they don't have a membrane surrounding their DNA. The other aspect of itself is that it's self-replicating. What is the difference between endosymbiotic and symbiotic? Oh, I see Opal also raised her hand. Let me answer Nisha's question, and then Opal, you can unmute and ask your question. So endosymbiotic is the, if you break down endosymbiotic into the endo portion, which endo means to engulf, well, endo actually means inner, but in this case for the mitochondria, they were engulfed by larger cells. And so they have this engulfing with also having a beneficial relationship with one another. For symbiotic just means that it has a beneficial relationship with each other, but it doesn't necessarily have to mean that it was within a larger cell. Like, you know how, I'm trying to think of another example. Because
like symbiotic, like so you have bacteria in your gut, and so also like called probiotics or probiotic or whatever, I don't know, but you have gut bacteria and you have bacteria in your gut, and it helps digest certain foods, and so you're benefiting the bacteria because you're giving it a place, you're providing it as an environment for it to stay, while the bacteria is mutually benefiting you by digesting certain, compounds and whatnot. So it's a mutually benefiting relationship.
Right. As Tim mentioned, yeah, the bacteria in the gut is a really good example. And also, it's not, the bacteria in the gut is not engulfed within like another cell. It's, just another organism that's lying around the gut. Does that help with the difference between? Okay.
I had a question, Janelle.
Yes.
You had mentioned mitochondria having its own DNA. And I'm not, this is a little bit beyond the lecture, but sometimes with those cold case files, when criminals are caught and they somehow use a parent and it's just the mother that has mitochondrial DNA. Is this going beyond the lecture, or do you want to touch on, or we don't need the MCAT?
I can touch on that briefly. Yeah. So
sure. Thank you.
So as Opal mentioned, for these DNA tests, for the mitochondrial DNA, they specifically usually look at the mother. The reason is because actually mitochondrial DNA is maternally inherited only. So all of this is just for, sorry, let me just double check this. Yeah, it's maternally inherited as mentioned as well. And so, you know, it's only passed down from your mom. And so that's why specifically if you're looking at mitochondrial DNA and you're looking at these cold cases, cold files, you would only be looking at the mom for that particular purpose because it's only passed down through one family member, one parent. Is that? Oh. Okay, great. Right, so I guess to continue with some of the points, we were up to the fact that mitochondria are self-replicating, right? So they have their own machinery actually to replicate themselves. They also have ribosomal RNA, tRNA, and proteins in the electron transfer chain and parts of ATP synthase just within the organelle itself. So it doesn't really require much of other, I guess, machinery. Sorry, just, okay, yep. And they also actually use a different system of transcription and translation versus the nuclear genes that usually are where most of the DNA is housed. And they have their own unique genetic code, which is really interesting. And these are all points that, may show that they were once prokaryotic. However, just a note is that although they have this ability to, create their own RNA and do their transcription and translation on their own, usually most of the processes are still done by the nuclear genes because it's just more cost effective for the cell to just do it that way. But yeah. And so to go into the next, the cellular membrane is, that semi-permeable phospholipid bilayer that surrounds the cell. And I just wanted to go into the different parts of the cellular membrane itself, just so it helps explain, why certain molecules go in and out of the cell. And the phospholipid bilayer, let me just see the pointer here, yes. So the phospholipid bilayer, because of bi, it has two of these phospholipid layers, and each of the phospholipids have a phosphate head group, so like the red dot, as you can see on the top, which is, and also has two fatty acid tails just right under it, the two yellow orange colored lines underneath the red head. And these are connected by this glycerol backbone to keep this whole structure connected. And the feature of the phosphate head group is that it's hydrophilic, which means that it is water loving. If you break down hydro as water and philic as loving, so water loving side, with also the hydrophobic fatty acid tails. And so if you break down hydro and phobic, hydro would be water and phobic is like, when you have a phobia of something, you're scared of it, you're afraid of it. So it's water fearing in this case. And these, phospholipids are amphipatic. And what amphipatic means is that it includes both this hydrophilic and hydrophobic portion. So both water loving and water fearing within this one structure. And another point I wanted to mention was that, so as I mentioned earlier about, why certain molecules have an easier time going through the lipid bilayer than others. Usually if a compound is small and nonpolar, that would be the fastest thing that can go in and out because of this fact that in the center area, right, the phospholipid bilayer is all filled with these hydrophobic tails. And so having this small and nonpolar allows it to move pretty quickly throughout to pass through the cellular membrane, as opposed to some other like larger and nonpolar or charged molecules. It just has a hard time with interacting with the cellular membrane phospholipid bilayer. And for these other molecules that do come in and out of the cell that may not be small and nonpolar, usually there are different mechanisms that allows it to go in. And so I'm just gonna talk a little bit about, what's embedded in the cellular membrane. And so it contains cholesterol, which is mainly used to improve the stability of the cellular membrane. So in situations where it's really cold and freezing, usually water turns to ice, so it freezes. And so what cholesterol does for the cellular membrane in this case is actually it increases the fluidity, so it allows it to still be fluid when it's in this really cold environment that would cause other things to freeze. And on the other hand, in really hot environments, usually it makes things very fluid. So, ice cube melts and it's just everywhere. And so what cholesterol does in that case is it actually allows the cellular membrane to be a little bit more stable and not as fluid as what you would expect in a warmer temperature, hot temperature. Also, within the cellular membrane are different types of proteins. And so there are integral proteins, which are usually embedded, as you can see in this center image. Integral proteins are usually embedded within the cellular membrane layer. Transmembrane proteins they usually span from the top to the bottom. I guess in this case, it looks pretty similar to the integral protein in this diagram. There are also peripheral proteins that are on the periphery, so usually facing the extracellular area. They can either exist on their own, or they can also be connected to another like integral protein, usually because integral proteins are usually embedded, so not necessarily passing sometimes through entirely the whole cellular membrane. And there are also carbohydrates that are along the cellular membrane sometimes, as you can see the dark blue areas on this diagram here. There's like this little portion on the right hand side where I'm circling with my pointer, and those would be the carbohydrates. Also, just to go back, these yellow portions would be the cholesterol that I was mentioning earlier. And right, so these different proteins help function in a way to allow certain molecules to come in and out. And sometimes, there are also protein channels, transport proteins that help regulate this. And the cellular membrane is a really important structure surrounding the cell because it does regulate what goes in and out. And sometimes you just don't want, and sometimes it can be harmful for certain things to enter the cell when you don't necessarily mean it to. And so that's why usually signaling is also involved in what goes in and out of the cellular membrane. Okay. And another thing I wanted to stress also is the difference between eukaryotes and prokaryotes, because that's also something that the MCAT likes to test on. And as I Oh wait, sorry, there's a question. Will the MCAT ask which amino acids are on the surface versus which ones are embedded? If so, which ones? So I guess this would go back to the amino acid lecture that Mark did a few weeks ago, but so they wouldn't directly ask which amino acids are on the surface versus which are embedded. Like the way that they will ask the question is kind of a little bit more complex than that. So, for the amino acids, they have different properties of being, non-polar, polar, or charged, right? Those are the three main categories that usually within the charged, it's acidic or basic. And so usually they will talk about the environment of the membrane, or like the extracellular environment or the intracellular environment, or also mention like within. And so you would in that case find the amino acid that best matches the properties that they describe. And so like something facing more of the extracellular environment, you would think of usually more polar, especially if they say something that's charged on the outside, if the cell is surrounded by something that's charged, or usually within the embedded inside the cellular membrane, as in this diagram, usually the hydrophobic tails. And so something to match that would be the non-polar amino acids. So that's how usually they would ask it.
Okay, so it's the like ones. So for example, the amino acids that are similar to the, the hydrophobic amino acids would be probably the ones that are inside versus ones. Okay, that's what I was getting at. Would they ask us which ones would tend to be inside versus ones outside? So it's kind of like attracts like.
Yeah, it's exactly like
Okay.
attracts like, yeah. And they will usually give you because the MCAT is multiple choice, give you the 4 different choices. And so from there, you can look at the amino acids they give you and see which one best matches
Okay.
the question.
Fair enough. Thank you.
Yep, no problem. Yeah. And so, yeah, if no additional questions, I'm going to go on to eukaryotes versus prokaryotes. And MCAT likes to ask this question in different forms, or they like to mention it within a passage. And so you kind of have to distinguish this. And so I wanted to stress a little bit on this. And so on the left side, you can see that's a eukaryotic cell. And on the right side, you will see a prokaryotic cell in this diagram. And the distinguishing factors between the eukaryotes and prokaryotes do not have a membrane-bound nuclei, nor do they have membrane-bound organelles. So in that case, you would usually see the DNA in prokaryotes to kind of just be like floating around. They wouldn't have a membrane surrounding it the way that a nucleus usually does in a eukaryotic cell. And they also don't have membrane-bound organelles. So you can see that they don't really have any of the mitochondria or Golgi apparatuses or endoplasmic reticulum. They do have ribosomes, which, as I mentioned earlier in the beginning of my PowerPoint, I made a slight mistake by mentioning that ribosomes are organelles, but they are not. They do not have a membrane surrounding them, so they are not considered organelles. And so, right, and in eukaryotic cells, they do have a membrane-bound nucleus, and they do have membrane-bound organelles. Another difference between the eukaryotes and prokaryotes is the way that they replicate. And so usually eukaryotic cells undergo mitosis for replication, while prokaryotic cells undergo something called binary fission, which they just like replicate off each other, as opposed to mitosis, which involves more complex structures, throughout the whole process. And another thing that I also thought would be worth to mention are the types of prokaryotic cell walls. So prokaryotic cells can also have cell walls surrounding their cell on top of having a membrane. And the two types of prokaryotic cell walls are usually the gram-positive ones or the gram-negative ones. And as you can see in this diagram, the gram-positive cell walls have a slightly, they do have a different composition between the part past the plasma membrane, right? So you see here that gram-positive bacteria are full of this peptidoglycan cell wall area. And they have like maybe a few strands of the lipoticoic acid, right? But then if you compare that to the gram-negative, they have one layer of this peptidoglycan here. And then they have lipoproteins, they have all these liposaccharides outside, forins, and they have this additional outer membrane aside from that peptidoglycan layer. And so the gram-positive bacteria, usually when they're looked under the microscope, they turn out to be purple. And I think this is mostly because of the peptidoglycan. It's just this, there's so much peptidoglycan that it's like saturated in color, as opposed to the gram-negative bacteria, which, they have this outer layer. So they end up turning this pinkish, reddish color, as opposed to the purple color. And so that is something that I wanted to mention. Also, viruses, they are not considered prokaryotes. The reason being is because they are actually considered intracellular obligate parasites. So they cannot, as you know, in usually a parasitic relationship, they cannot multiply just by themselves. Like they have to depend on another cell's machinery in order to do all the processes that it wants. And if it doesn't have a cell that it's kind of leeching off of, then they kind of just die on their own after some time because they just can't do those processes on their own. And so I wanted to end with another question just to test that. I'm also going to give 2 to 3 minutes.
Okay, it looks like most people voted.
Okay.
80% voted D, 20% voted A.
Okay. Okay, so I'm just gonna release the answer. So the answer is D. So the majority of you guys got it correct. So I guess the other 20% said the organism has a cell membrane. So let me repeat the question. It says a scientist observes a new organism through a microscope. Which of the following observations tells her that the organism is eukaryotic? And if I'm just gonna go back a little bit in the slides. And so, one of the things I really wanted to stress was that prokaryotic cells do not have membrane-bound nucleuses and the nuclei, and they also do not have membrane-bound organelles. And so if you actually look at the type of prokaryotic cell walls here, this example, it actually shows you that these prokaryotic cells do have they can have a plasma membrane outside of their cell. They can have like a plasma membrane and then also another cell wall on top of that. So that is why in particular for this question, well, for all, reasons of why a cell would be eukaryotic is that it has this membrane-bound nucleus. That is really a big defining feature of what makes a cell eukaryotic. And so, yeah, thank you guys for all attending here. I know it's Mother's Day, so if you guys have, Mother's Day festivities, feel free to head over and spend time with your mom. If not, I am here for any additional questions. If you have, questions about biology or even psychology, sociology, because usually I think I will be I am a MCAT tutor that will be mostly focusing on biology and also psych-soc in this program. And so, yeah, and we will also try to get the PowerPoint recording to you, and we'll share that information on GroupMe or other Socratic Med outlets. And Opal, yes, you raised your hand.
Yes, with the peptidoglycan cell wall, the gram-positive. There were questions. I think I took one of those full length, which had to do with bacteriostatic versus bacteriocytic. Could you explain that in terms of the cell walls and what's the difference between bacteriostatic and cytic?
Bacteriostatic and cytic?
Yeah, like penicillin, is it a bacteriostatic or is it I'm trying to figure what the difference is. Do you know anything along?
Hold on, let me just I'll share for now.
I've never heard that.
I've never really heard that. Could you actually go more into like what the?
I'll send the question. I could send it to the office hours. It's okay.
Or if you could like also generally say maybe what the passage was about.
Oh, I'm reading it right now. It says bacteriostatic agents are capable of inhibiting the growth of reproduction. Inhibiting the growth or reproduction of bacteria. Different from bactericidal, capable of killing bacteria outright. So bacteriostatic, you can break that down. Static means to like halt or to, it's not moving.
It's
so
the same.
Yeah. And so that's inhibiting the growth. You can kind of put those two words together.
Yeah, also I wanted to mention the bacteriostatic and acidic, they're not actually bacteria themselves. It's agents like chemicals or other compounds that actually work on bacteria to like inhibit them or just kill them or inhibit them. So like kind of like antibacterials basically.
So they're not the cell walls. That's okay.
No.
Yeah, okay,
those are agents. Yeah.
So penicillin, would that be a static or acidic? I think that was what the question was getting.
So penicillin, and actually, as you may know, penicillin is a very common antibiotic. It's an antibacterial medicine that we give to people who have like bacterial infections. Yeah, and I don't think they would expect you to know that. Likely, if it was tied to a passage, I think that's something that
They'll give us.
they would tell you in the passage, they would give you some hint in the passage as to how to solve that, because I don't think they would expect you to know that.
Sure, I'll find the passage that
Yeah.
I go over.
Opal,
Sure.
I just looked it up. It says it's bactericidal because it inhibits bacterial cell wall synthesis.
So it inhibits, so it's kind of static. It's not killing it, it's just preventing growth.
Well, it's preventing the cell wall synthesis, which it's not. I believe they said bacteriostatic is directly inhibiting the growth. So bacteriostatic agents are likely targeting like the DNA, some kind of, or perhaps the RNA, maybe even protein production.
Yeah, so I would assume bacteriostatic would focus more on kind of growing, so like DNA replication or RNA production. Protein synthesis might actually like be cyto, because it might kill if you don't have any protein. So I think DNA replication is more likely to be the target of bacteriostatic.
Now, Opal, they would give you a passage. They wouldn't expect you to know that.
Yeah, it wasn't a passage to be. It wasn't a passage, and they kind of defined what was going on and asked what would penicillin be. I think the question was, is penicillin acidic or static? And it was passage based. So
yeah. Well, I mean, it's good thinking, like connecting, thinking a bit more in application terms. Even if you don't have to memorize bacteriostatic, it's good to be thinking like that.
So Janelle, going back to that last where are these agents acting on that the cell wall that you showed for the bacteria? Where are they acting?
Sorry, let me go back.
And that was my last.
Oh, like on this diagram?
Agents, yeah, where would they be acting?
So as Mark mentioned, I think it would probably target trying to get to the DNA replication site. So actually would try to pass through the cell wall, pass through the cellular membrane, and then go pass through also the nuclear envelope to get to the nucleus in order to stop that kind of process.
And then get to the, okay. Yeah, okay. Fair enough. Thank you.
Yeah, of course. Yep. So if anyone else has any questions, feel free to put in the chat or raise hand, whatever you guys want to do. Or if you also have to get going, that's completely fine too. Should we, I guess, should we?
Yeah, I think we can wrap this up. If no one has questions, if you have questions, you can always ask in the group meet chat, and we'll save them and cover them in one of the lectures. So.
Right. Yeah. Just to stress, if you have any questions that you want to ask me, you can feel free to message me via group meet. I'm in the group with you guys, so you can just find me and send a message to me. And then also that we have office hours every Saturday from 12 to 2. If you guys have any other questions during your MCAT studying.
All right, guys. I'm gonna end this meeting. I'm gonna end this call. All right. Everyone, have a good day. Happy Mother's Day.